Recent headlines have drawn attention to a remarkable scientific claim. Researchers have discovered a kind of kill switch within the human body that, if activated, can selectively destroy cancer cells. While the term kill switch may sound dramatic, it accurately reflects what scientists are beginning to understand. It's a natural self-destruct mechanism built into our cells which, if properly triggered, could revolutionize cancer therapy. The promise of these discoveries lies in their ability to kill cancer cells without damaging healthy tissue, offering hope for more effective and less harmful treatment options. Just a reminder to hit the like button and subscribe if you enjoy this video. It won't cost you anything, and I would appreciate it very much. Thanks. Now let's get started. Between 2023 and 2025, key breakthroughs have been made by multiple research teams. In October 2023, scientists at the University of California, Davis, published findings that highlighted a precise targeting method for a known receptor involved in programmed cell death. This receptor, CD95, also known as FAs or the death receptor, has long been known to trigger apoptosis, or cellular self-destruction. What was new in this study was the ability to activate it with high specificity, particularly in cancer cells. This type of targeted triggering could pave the way for safer, more refined treatments, especially for solid tumors where current therapies often fall short. Building on this momentum, researchers from the Jackson Laboratory and UConn Health announced another significant development in early 2025. Their work focused on the TRA2B gene, which plays a role in RNA splicing, a process critical for the proper functioning of cells. They found that cancer cells tend to silence poison exons, which are segments of RNA that act like natural off switches for certain proteins. By restoring these poison exons using synthetic RNA fragments known as antisense oligonucleotides, or ASOs, these scientists were able to trigger the kill switch and cause cancer cells to self-destruct. This method showed particular promise in laboratory studies involving aggressive and treatment-resistant forms of cancer. In addition to exploring natural cellular processes, scientists have also been developing synthetic and engineered approaches to induce cancer cell death. At Stanford, Northwestern, and international labs, researchers are designing protein-based tools. These tools force cancer cells into apoptosis by disrupting the balance of proteins that normally keep them alive. For instance, one method involves gluing together cancer-driving proteins such as BCL6 to activate genes responsible for programmed cell death. Other efforts focus on constructing synthetic biological circuits, engineered systems that act like Trojan horses. These circuits can be inserted into cancer cells using nanoparticles or viral vectors, where they lie dormant until triggered to initiate self-destruction from within. These discoveries represent an important shift in how we think about treating cancer. Instead of broadly toxic therapies like chemotherapy and radiation, which attack both healthy and cancerous cells, these strategies aim to target only the diseased cells. This precision could drastically reduce side effects and improve the quality of life during treatment. Moreover, they offer new hope for cancers that have been especially difficult to treat with conventional approaches, such as certain brain and breast tumors. How these kill switches are delivered depends on the specific technique. Gene and RNA-based therapies, such as those involving ASOs, are likely to be administered through infusions or targeted injections that deliver the material directly to tumor cells. Protein-based treatments, such as monoclonal antibodies or engineered death activators, may be given intravenously. Another approach involves immunotherapy, where a patient's immune cells, specifically T cells, are extracted and genetically modified in a lab. They are then reinfused into the body to hunt down and destroy cancer cells. In cases where synthetic gene circuits are used, delivery might involve viral carriers or nanoparticles designed to penetrate the tumor and trigger its collapse from within. Despite the promise, these therapies are still in the early stages of development. Most are being tested in laboratories or animal studies, with a few beginning initial clinical trials. Before they can become standard treatments, they must go through several phases of testing to prove they are safe, effective, and deliver consistent results. If progress continues without major setbacks, it's possible that some of these therapies could become publicly available by the late 2020s, though broader adoption will likely take longer. However, it's important to remain realistic. While these mechanisms show great potential, there is currently no universal cure for cancer. Each type of cancer behaves differently, and what works for one may not work for another. Moreover, cancer cells are notoriously adaptable. They can mutate rapidly, evade immune responses, and find ways to bypass treatment. Translating laboratory successes into real-world therapies is a complex and uncertain process. Even so, components of these kill switch mechanisms are already being explored in some advanced treatments. The CD95/Fas receptor pathway, for instance, is under investigation in clinical settings and plays a role in some immunotherapy strategies. Engineered T-cell therapies like CAR-T already use aspects of kill switch logic, though they are currently limited to certain types of blood cancers. To summarize some of the most recent and promising discoveries, the CD95/Fas receptor can now be targeted more precisely to initiate apoptosis in tumor cells. The TRA2B poison exon mechanism shows that restoring suppressed RNA segments in cancer cells can trigger self-destruction. Engineered protein gluing techniques can rewire cancer cells to activate death genes, and synthetic genetic circuits can be embedded within tumors to act like hidden bombs that go off only under certain conditions. These approaches are still experimental, but they represent important steps forward. In conclusion, scientists have made real progress in uncovering and reactivating the body's natural kill switches for cancer cells. These discoveries open new avenues for treatment that could be more precise, less toxic, and more effective than current methods. While we are still several years away from widespread clinical use, these findings mark a hopeful turn in the long battle against cancer. Not every cancer will respond the same way, and more research is needed to fully understand the risks and benefits. Still, the idea of harnessing the body's own mechanisms to fight cancer represents one of the most exciting developments in modern medicine. Before you go, check out some of my other videos. Don't forget to hit the like button and subscribe. Thanks for watching. We'll see you at the next one.